Good evening, ladies and gentlemen, journey through the world of next-gen sequencing. I am Sakshi and we are thrilled to have you with us today for the special workshop. Let's dive right in. We, the Dr. Omics Labs Private Limited, are passionate about enhancing knowledge and understanding in the field of genomics. Our mission is to empower individuals like you to explore the vast potential of next-gen sequencing. To guide you on this incredible journey, we have two remarkable experts with us. Please join me in the welcoming Dr. Yogesh and Ms. Dhanashree ma'am. They bring a wealth of knowledge and experience to the table. Today, you will gain more knowledge about why NGS matters, how to do planning for your NGS research. You will do practicals with data. We are sure that you will enjoy the sessions, and once again, welcome to all. Thank you for joining us. So first of all, I am welcoming Dr. Yogesh for the first session. Dr. Yogesh, an accomplished scientist with a BSc, MSc, PhD in bioinformatics, currently serve as a senior PDF scientist specializing in biological data analysis. His expertise spans across various domains, including scRNA and RNA bulk analysis, genomics, proteomics, transcriptomics, NGS data analysis, and machine learning. Dr. Yogesh is actively engaging in multiple projects centered on cancer research with a primary focus on unraveling the interaction of the tumor, microenvironments, and T-cell plasticity. His work also involved predicting responses to immunotherapy and chemotherapy, distinguishing between responders and non-responders. Dr. Yogesh's commitment to advancing our understanding of complex biological systems is truly remarkable. So without wasting time, I am hand over to the Dr. Yogesh. Thank you.
Yeah, thank you so much for the introduction. And so let me share my screen first of all. So I hope you are able to see my screen. Hello. Yeah, thank you so much for your introduction. And hello, everyone. And I welcome all of you to this session of understanding the NGS and bioinformatics together at the same platform. So I would like to say good evening, and again, thanks for inviting me this talk. So in next 1 hour, I'm going to give you some highlights about the NGS technologies and about how you can use the NGS in different field, biological field, and also some other fields. And after using the NGS, how you can use the bioinformatics to analyze the data which was produced by the next generation sequencing platform. So the topic will be transforming healthcare and research with NGS and bioinformatics. So now you see this picture. It's a beautiful picture where I work. It's a picture of Hamburg. And here is my university where I was working as a senior postdoc scientist. It's called University Medical Center, Hamburg University Eppendorf, which is in Hamburg. So I'm working here since 2020. So now, without wasting any other time on this, I would like to come directly to the presentation. So in next 1 hour, we are going to understand this revolutionizing bioinformatics with next generation sequencing. So as you know, that the future of NGS and bioinformatics is now. And the future of this NGS and bioinformatics has been more exposed after the COVID-19 pandemic since 2019-20, and now it's a very booming field in the life sciences. So now first, I'm going to tell you about some basics introduction of next generation sequencing. So what is actually sequencing? So sequencing in the context of genetic and genomic refer to the process of determining the order of nucleotide in a DNA or in an RNA molecules. So basically we have two types of sequencing. One is DNA sequencing and another one is RNA sequencing. So now what is DNA sequencing? So this is the most common form of sequencing, and it involved determining the order of nucleotide in a DNA molecule. And DNA sequencing is very crucial in various fields, including like genetic, genomics, and molecular biology. And it allows researchers to study the genetic code, organism, identify genes, mutation, genetic variation, and to understand the molecular basis of some of disease. Then another form of sequencing is RNA sequencing, or we also say it RNA-seq in the short form. So it is a very specialized technique for determining the sequence of nucleotides in RNA molecules. And it is particularly valuable for understanding gene expression patterns and can provide insight into how genes are regulated and which genes are active in a specific biological sample or in a specific biological condition. Okay, so it also helps in identifying and diagnostic biomarker for initial experiments. So coming very briefly to the brief history of sequencing. So until the starting of the generation of sequencing and until now, we have started for the first generation, second generation, then we come to second generation, and now we are very advanced and we are coming to the third generation sequencing. So not going more deeper into this sequencing, I would tell you more very briefly about these techniques. So as you have maybe you have learned about these sequencing methods and about us like basic type of sequencing. All of you have heard the name of Sanger and Maxon Gilbert. It's a very basic sequencing method which was started for the first time. And that sequencing method is categorized as a first generation sequencing. And that method is able to sequence about 100 to 1000 base pair fragment at a once. And at the time of, then we move to the second generation sequencing. And in the second generation sequencing, we have more automated platform, which now use high throughput and use parallelization of sequencing reactions, and they are able to sequence like 50 to 500 base pair fragments. And now is the third generation sequencing, which is fast as well as used for long reads. And the machines of this third generation were very compact. It is called PacBio Oxford nanopore sequencing, and these method was used for sequencing a long read. So now coming to the beginning and booming of the NGS sequencing. As I told you that the first generation sequencing was completely based on the Sanger and Maxon Gilbert method. And also this second generation is also on the same basis, but the first generation sequencing was used for very long reads, like 500 to 1000 base pair. And that sequencing method was automated in 1990 when the Human Genome Project was started. So this technique and technology of NGS, sequencing the DNA and sequencing the RNA, have been developed over, we know that more than 50 years, but the revolution came after the Human Genome Project was started. So since completion of the Human Genome Project, the Human Genome Project was completed in 2003. So they to sequence the whole genome of human, they automate the Sanger and Gilbert Maxon Gilbert method, and then they sequence the whole genome of human. So since completion of that project, the speed of sequencing has increased, and also the cost has been also decreased. So around overall, like this Human Genome Project was completed in several billions of dollars. But today, if you will go for a sequencing, you will cost some thousands of dollars. So this is the revolution of sequencing from the first generation until date. So the question comes in our mind is that why we need sequencing? So because So sequencing is a fundamental tool in modern biology and has a wide range of applications that impact human health in different fields like agriculture, environment, scientific knowledge in general. So there are several, I categorize, there are more fields, but I categorize some of the fields over here like academic research, you use sequencing, next generation sequencing and sequencing methods. You in clinical research you use, in hospital applications you use, in biopharma, biotech and biochem laboratories. So there are several other fields where you need the sequencing. So first field is like to diagnose a disease, you can do the sequencing. For drug development, you need the sequencing. And to understand the genetic information, you need this. To in the field of forensic science, in the environmental monitoring, in microbiome analysis, you need to do the sequencing. To study the evolutionary relationship between the different species, you have to do the sequencing. In cancer research, of course, in cancer research, it's nowadays very common to do the sequencing and to study the cancer. Then in also historical and anthropological research, you need to do the sequencing. And species conservation, to understand the species conservation, sequencing is must. In agriculture and crop improvement, then in basic research, and also in petroleum industry. So you many of you might be very like surprised to hear that how we use the sequencing in petroleum industry. So one of my friend, they are working on this type of project, and the project is based on like these refineries wells from where they mine the oil. So once you complete digging all the oil from the well, so there are still possibility left that there must be some more oil in those wells, but that could be very hidden in the very hidden form. So they isolate the samples from those wells, and then they try to identify that what are the microbiome which would be able to like degrade or form these oils. So this was very interesting study. So here in this study, they use like sequencing, they sequence the microbiome, and then they identify some of the microbiota in the microbiome in that refinery oil well samples. So you can see that there are many fields where you can use sequencing. And now next come that once you use sequencing, you have to do the analysis of those sequencing. And for doing the analysis, you need the bioinformatics skill and you need the bioinformatics knowledge. So what we can sequence, actually, then the question comes here that what we can sequence using NGS method. So first of all, as you know that we can sequence the whole genome and then we can also sequence the whole exome. So exome is the protein coding region of within the genome. Then we can also sequence transcriptome, which is RNA sequencing, epigenome, which is used to study the epigenetic modifications such as DNA methylation and histone modification. Then we can sequence metagenome, which is associated with microbial communities. We can take a sample from any of the soil, water, and human body or human gut, and we can identify or sequence this metagenome, these different microbial communities, bacteria and fungi. Then we can also sequence metatranscriptome. So metatranscriptome is a combination of mixed type of microbial communities. And we can also sequence mitochondrial genome, mitochondrial DNA, which is separated. This mitochondrial DNA which is separated from the nuclear DNA and can be sequenced to study evolutionary relationship and access mitochondrial disease. Then next, what we can sequence is cancer genome. So NGS is crucial, it is very crucial in cancer research for identifying mutation and genomic alteration associated with cancer development. and its progression, and also for drug response. So next, we can also sequence the single cell genome, which is very these days, which is very booming and very famous, you can say. And the single cell sequencing allow the study of individual cells. So we can sequence not only the whole RNA, but we can sequence a single cell from the samples, and we can understand the diversity in the cells, and we can properly understand the cell development and heterogeneity in tissue. Next, we can also sequence epitome of the so for epitome sequencing. So epitome is used to study the immune response, and using the next generation sequencing, we can sequence the variable region of antibodies, and like T cell receptors, those receptors which are present on the T cells. And by sequencing this, we can understand the immune system and its function. Then we can also sequence chromatin accessibility. So chromatin accessibility is also called as attack sec and DNA sec, and it can be used to sequence open chromatin region, providing insight into gene regulation and enhance the activity. So we can also, next we can sequence structural variants. So using this NGS method, we can identify structural variants such as insertion, deletion, or duplication and inversion in the genome, which play an important role in genetic diseases. Next, we can also, of course, we can also sequence viral genome, either it is RNA virus or DNA virus. We have seen this example very much in this COVID-19 pandemic. Then we can also sequence ancient DNA. So next generation sequencing allow the sequencing of DNA from ancient species. If you get some of the samples from the ancient mummies or some other organism, you can also sequence that. And next is you can sequence environmental DNA. So DNA from any environmental sample, from soil, from water, or any other, from plant, any. And next we can also sequence synthetic DNA. So synthetic DNA is a DNA synthesized in the laboratory for synthetic biology application. When we do the gene editing, and biotechnology can sequence to confirm that integrity of the design sequences. If the DNA, if you do the gene editing in the DNA, synthetic DNA, then you have to do the sequencing, and you can confirm that if that editing is fine or not. So now next question comes up, how this work, how the NGS is working, and what are the different steps for the NGS. And at the end of doing the NGS, what we have to do what we have to do of this data. So NGS is majorly divided into four. NGS is basically divided into three parts, and the fourth part is the analysis of the data which you get from the NGS sequencing platform. So first step is preparation of sample. So this first step is very common step, which for you need to prepare the sample for doing the NGS sequencing, no matter if that sample is of microbiota, bacteria, fungi, or that sample is of any virus, bacteria, or human, mice, or any of the samples. So you have to do the sample preparation before going to the NGS workflow into the NGS. Then after doing this sample preparation, next step is the library preparation. Either it could be if you are sequencing RNA, then it will be a RNA library. If you are sequencing DNA, then it will be a DNA library. So second step is library preparation. And third step will be the sequencing. So you have to give your whole preparation and libraries to the sequencing machine, and like sequencing platform, and then it will sequence whole sample for you. And after completing the third step, you will get the data in the form of a result. And then you have to proceed with the bioinformatics analysis to understand the sequencing and to understand the sample and to study more deeper about those samples which you have sequenced. So there are basically one file, different file format you get from the sequencing machine. So the very common one is the FASTQ file format, which you get as an output of the sequencing machine, no matter what platform you are using for sequencing. It could be a Illumina, it could be a Roche, it could be a 454 or any other platform. So the file format you will get is the FASTQ format. Then you have to do the pre-processing and some other step, which my colleague will tell you in the next step, in the next talk. So once you get the sequencing data from the sequencing machine, then you have to do the bioinformatics analysis. And I will tell you a very little bit about the file format, how you will see what you will see in the file which you get from the sequencing machine. So understanding the output file format of next generation sequencing. So the first format is the FASTQ format, which you get directly as an output from the sequencer. So here you can see there are 4 lines of sequence. So this is a very small chunk part of the sequence. It is not the whole sequence. So its length of the sequence is 76. So you can see these nucleotide, they are 76 in number, and you see the CT, these are the nucleotides, and at the end you see N, which means that they don't detect this at the time of sequencing. So I will tell you more briefly about this sequencing file. So the first line you see is the header line, which begin with the @ sign, and it's labeled as sec ID. So here is an ID of the sequence. Then the second line is contains the nucleotide sequence, which you can see over here. And the third line is assigned with a plus sign. And this line also have this ID and also give you the information about the length of the sequence, which is 76. And the last line, which is this, it tells us the quality score of the sequencing. So next, once you will get the FASTQ file format sequence from the sequencing machine, then you run the alignment. And so the background noise is coming. I think someone have to mute himself. Yeah, it's fine. Thank you. Thank you. There is a noise, but I will continue. Sorry. So once you get the FASTQ format file from the sequencer machine, then you have to do some other bioinformatics step. And the number one step would be the alignment of the data set. So once you get the data in FASTQ format, then you go for the alignment of the data. If the data is from the mice, then you have to align and map that sequencing data with a mice genome. If the data is from human, then you have to map that sequencing output sequence with a human genome. So there are several tools for doing this. So most common tool is the star alignment tool, which you can use for aligning this sequencing with your reference genome. And once you completed this alignment process, actually this alignment process takes several hours to several days. It depends upon the computational power what your machine have. So after doing the alignment, you get some of the file which are called SAM file, BAM file, and also the output result file, which if you are using the RNA sequencing, then you will get some of the read file, a count matrix file, and also the SAM or BAM files with that. So actually, what is the SAM and BAM file? So SAM and BAM file are the primary used to store information about the alignment of sequence read. So they have some information in their sequence read. So such as those generated by high throughput sequencing technology. So I'm going to tell you some of the information present in the SAM and BAM file. So you can see here the different section, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10, 11, 12. There are at least 12 different sections. This is an example of the SAM file. So first section where you see this is, it start with some of the header line. It's the name of this sequence. And then you have number 16 that indicates the bitwise flag field represent various alignment properties, what properties you have used during the aligning these sequences. Then you have the chromosome, reference chromosome, what reference genome you use, and what is the chromosome of the genome which was aligned to this sequence. Then you have mapping position of your sequence, query sequence, to the reference genome. It is indicated as 1000 here. Then 30 is the mapping quality, which is good. And then we have 50M, which is the string which indicate that you have 50 base matches with the reference genome. Then you have star and zero sign over there. Star and zero are placeholder for field related to pair and data, which are not used in the example. So then we have TTAGG, some of the chunk of the sequence. So this is the sequence of that read. And next we have star indication over there. So star represents just a placeholder for any optional field. Then at the end you have some of the XTAR. So this is also again the optional field indicating the alignment was reverse complement. So this sequence you might be like curious that this is very small chunk, but it is bigger one. It is bigger, but I have represented a very small sequence over here. So this is the second file format you get after doing the alignment of the output file of the sequencer. So once, and with these BAM and SAM file, you will also get the, if you are doing the RNA, you will also get some of the these expression matrix, RNA expression matrix file. So now next would be the next generation sequencing. What is the market trend and what is the future of next generation sequencing, and also the bioinformatics. So this is a data which I have collected from CAGR, which is a global next generation sequencing market. It represents that at the moment, the market value of next generation sequencing is 7.7 billion in 2023, and it would be expected to be 28.47 billion until the end of 2033. And if you compare the market of bioinformatics, so it says that until 2019, the market of bioinformatics is 10.73 billion. And until end of 2026, the market of bioinformatics would be 37.03 billion. But now I expect more. Personally, I expect more, that market of this bioinformatics until the end of 2023 will cross $40 billion US dollar. Because now the demand of bioinformatics is very much in the market of biotechnology and healthcare centers. So now the question comes as that, okay, what I have told you is a composition of doing some experiment. And if you did some experiment, then you send your samples, prepare sample to the sequencing. And then once you get the sequence from the sequencing company, you will get it. And then only you have to analyze those samples. But what are the alternative? Because sequencing process is a costly process and everyone cannot afford it. So now the question comes here, where we find the raw data and how to work with available online data. If someone wants to work on the sequencing data and they don't afford to do the sequencing, then they can practice by themselves. They can do some of the bioinformatics analysis by themselves, and they can also make those analysis in the form of paper, and they can also go for publication of those papers. So there are several databases where you can find out these raw data set, which you get directly from the sequencing, different sequencing platform. So the first database is SRE database, which is Sequence Read Archive. So this database contains the very raw format of the sequencing data. And another database is GenBank. GenBank is another source which was under NCBI, and it allows you to submit RNA bulk sequence, metagenomic, and it's a database of genetic sequence and associated metadata. Then third database is MGRAST. It is a metagenomic rapid annotation using subsystem technology. It is a very specialized repository for metagenomic data only. Then there is another one is called European Nucleotide Archive, which is called ENA. And this is a primary repository for nucleotide sequence data in Europe. So it accepts bulk RNA sequence and metagenomic data submission and making it a suitable choice for research in Europe. So this database is specifically for European countries. Then we have DDBJ, DNA Data Bank of Japan. So this is also one of the 3 primary nucleotide sequence database. And along with GenBank and ENA, researchers can submit their raw data, including bulk RNA sequence and metagenomic of DDBJ. Then the 6th one is Genome Expression Archive. So it is a part of European Bioinformatics Institute, which you call EBI, and specialize in gene expression data, including bulk RNA sequence, and it allows researchers to archive and access gene expression data. Then the 7th database is National Genomic Data Center, NGDC. So this is based in China, and NGDC provide data resource for a wide range of genomics research, including bulk RNA, metagenomic, and any researcher can submit their data over here as well. So these are the several databases and several resources where you can go and find out the raw sequences of the published papers. You can download those sequences, the raw sequences. Sometime in some of the database, you get the expression matrix. If there is an RNA sequence, suppose there is a publication where they have done a research and they compare like 2 conditions. For example, if they compare the 2 condition, healthy and disease condition, and they perform to compare those different 2 conditions, they did RNA bulk sequencing. And sometime what they do, they just including the submitting this raw data sequence. The raw means the one which they get from the RNA from the sequencer machine. They also submit the expression matrix in the database. So I will show you some, my colleagues will show you some of the example in next talk. So in that case, if you are lucky and if the author has submitted the RNA expression matrix data in any one of these database, then you can directly download the data and you can just run the different pipelines for doing the analysis using the bioinformatics tool. And if there is a case that you don't find the expression matrix data, then you can download the raw data set. Then you can use the bioinformatics tools for doing the alignment. You have to do the alignment of the raw sequence with the genome. If it's a mice, then you have to do the alignment with mice. If it's human, then with human. So now dealing with the next generation data set. So I will tell you one example. I have put it one example. This is a paper which is recently published. We published this paper in month of August this year, and this is very interesting paper where we have revealed that short-term dietary changes can result in mucosal and systemic immune depression. So if you change, so the purpose of this main objective of this research is that if you change your diet, not even for the longer term, but if you change your diet even for the shorter term, then you can depress your immune system. So for example, in your right side, you see this graphical abstract. In this graphical abstract, you see that we feed our mice, set of mice with a regular diet and with a feast diet, means fat-rich diet. So we feed these mice at the day 0. We collect the sequence or sample from these mice. Then we feed these mice with a fat-rich diet. And after 3 days, we collect the microbiota as well as the sequence as well as the different other samples from these mice. Then again, we feed the same mice with a fiber-rich diet. And after 6 days, we collect the sample. Then again, we feed these mice with a fat-rich diet for the next 3 days. And then at the 9th day, we collect the sample. And then we reverse the diet of the mice to the fiber-rich diet. And then at the end of 12th day, we collect the sample and we sequence all these samples. So what changes we get were very interesting, that we see the oscillation of changes in the genes. Over here, you can see the day 0, the genes which are high up, like showing high expression in the mice, they then, when the mice was switched to the fat-rich diet, these genes going down, they are showing low expression in the mice. Then we again switch back these mice diet, they go up, then they go down, and then again they go up. And similar case was observed with the microbiota over here. This is the gene, sorry, these are the gene which you can see the oscillation. We see the two pattern of genes. Pattern 1, there are several genes which are first showing high expression in day 0, then they go down. And there are some genes which are low in the day 0 and they go high in the day 3. So similar pattern we have also seen in the microbiota with the bacteria which were present in the gut. So what we have done, we collected the samples, we did the sequencing, RNA bulk sequencing, we collected the sample, we did the sequencing for the microbiota, for the gut bacteria which were present in the stomach of these mice, or you can say the gut of these mice. And then we did the bioinformatics. Using the bioinformatics tools, we perform the sequence analysis and we look these observation. And then we come to know, okay, there are several genes, there are several genes which are go high with a when you consume the fat-rich diet, and there are several genes which go low when you consume the fiber-rich diet. So now, why I have shown you this example, it's because, so after doing this whole analysis, after doing this whole sequencing and bioinformatics analysis, so we have submitted our raw data in different databases. So you can also see, you can go to this paper and you can see the section in the paper which is called data availability. So under this section, you can see there are some of the databases or some of the different resources where we have submitted our raw data set in these databases. So first one is European Nucleotide Archive. And another one is Sequence Read Archive. And the next database is Gene Expression Omnibus, which is called GEO. So first one is the GEO database. You can see that we have submitted all the raw data sequences over here. And this is another database called European Nucleotide Archive. We have also submit our data over here. Then we also submit our data in this database. So what the purpose of telling you all this is that, so even if you don't have experience with the handling, if you don't have like resources to sequence your samples, and your experiment some more like less or more is correlating with the experiment which was previously published, then you can also download the data set which was published previously, and you can identify if you're lucky, then you can also identify some of the genes which meet your objective of research. So without doing any sequencing, you can, in this way, you can also do the sequence analysis using some of the bioinformatics tools. So now what are the, I will talk about a little bit more about the career opportunities over here. So what are the career opportunities if you have a skill of doing the bioinformatics, NGS, especially the NGS analysis? So one thing I would say that doing this bioinformatics analysis is a very tricky task and also a very laborious task, because you have to deal with the different programming language like R, Python, Java, or maybe JavaScript, or whatever language you like. So I suggest you should only start the bioinformatics if you want to make a difference in people's life, if you able to solve the problems, and if you are ready to take challenges. If you have patience, then of course you go for the bioinformatics. Working as a part of a team, if you like to work as a part of a team, then it's good choice to be a bioinformatics or doing the sequence and just sequence analysis. If you also like to work independently, you can also go you can choose the bioinformatics and go further. And the most important thing is that if you like to help people when they need help the most, then you choose this bioinformatics. So because a bioinformatician or doing a person who is doing the next generation sequencing analysis, they don't very oftenly go to the wet lab and they don't perform the experiment. They have to do, they only sit in the office and do all the bioinformatics. And the challenging task for bioinformatician is that they have to be involved in multiple projects. So these are the things, if you enjoy these things, then this doing the bioinformatics is very good field for you. So now what will be the designation if you do the bioinformatics? So you can be a bioinformatician, you can be a bioinformatics analyst, you can be a bioinformatics scientist, computational biologist, bioinformatics engineer. So the difference between these different all the fields is very thin line. So if you being a bioinformatics analyst, then you will do only the analysis part. If you are doing if you are bioinformatics engineer, then you will be dealing with the development of different software, different pipelines, and you can be a genome analyst, biological data scientist. And if you are a biological data scientist, then you have to deal with programming and like everything. So now, as I have told you that economy of this NGS and bioinformatics is very fastest growing in the field of life sciences. And these two fields were growing currently, and the future of this field is very bright. And at the moment, you have a lot of job opportunities if you have a knowledge of bioinformatics and knowledge of NGS data analysis. And there is a job security, of course, if you have these skills. And you have a flexibility also when you are choosing this as a career. And I would like to tell you this is very rough estimate of salary if you are a bioinformatician, or if you are a computation biologist, or you are a biological data scientist. So salary range at the moment in USA starting is from $70,000 per month annum to $200,000 per annum. So this is very basic like salary. And the good thing is that in the field of life sciences, if you are a bioinformatician or a computational biologist, you can earn more than the regular biotechnology person or microbiology person. So your salary will be higher if you have these skills. So what are the job offering in this field? So I did the very random search to show you one example. I did this search in the LinkedIn. If you will see, if you will do a search as a bioinformatics and you will set the location as Germany, because I set Germany because I'm working in the Germany. So you can see there are 1,763 job openings only in the which were advertised in the LinkedIn job opening for the bioinformatics. And in the USA, there are 1,036 job opening which are present in the LinkedIn platform. There are different several other platform which where they publish the job opening of computational biologist or bioinformatician. So this is just a basic example to just tell you that these two fields, these NGS data analysis or bioinformatics, bioinformatician, these two fields were like booming field at this time. If you have a knowledge of doing this analysis, NGS analysis, then your demand is very much in not only in the USA or Germany or Europe, but all over the world, also in the India. So there are some of the basic, some of I have listed few of these job offering companies which are very famous in the worldwide, like Lumina. They offer the bioinformatics job, Thermo Fisher, Quigen, Roche, and Merck, and Novartis, Pfizer. You must know this. And Evotech, 10x, IBM, and there are several, like hundreds of other companies which offer a job to the bioinformatician or computational biologist. So here I would like to thank you, and I would like to end my presentation. So this is my small group. I'm working with all of them. And these all people were the clinician scientists in this hospital. So I'm the bioinformatician over here. There is one more bioinformatician working with me. And as I told you that being a bioinformatician is also a very challenging task because you have to deal the multiple projects. So I was, and at the moment I am involved in all of their projects being as a bioinformatician. So I would like to end up here. If you have any question, you can ask me and you can write me. Thank you so much.
Hello.
Yeah,
Am I audible?
yeah, I can hear you.
Yeah, this is Kirtana from India, and I have been pursuing master's degree second year in human genetics.
Okay.
I was I am being an intern in TCAPSILA presently and doing these learning these molecular biology techniques and NGS too. Had finished the course. So, how to get into the industry?
Okay. So you have finished your master's, right?
No, still not yet. There is one sem left.
Okay. So I think it would be better to finish up the master's and to get a internship in a very nice either institute, or if you are lucky, you can get the internship in the any company. The after uh, actually I can say that internship is the most key part to go further either in the industry or in the research area, or if you want to do the further study. So in doing the internship is the important part over here. So if you will join the internship in any of the company or in any of the research institute, then it will be a opening door for you to proceed or to go with the industries.
Can and I'm in third sem, and in the fourth sem we had this idea of project building.
Yes.
So I want to go through this NGS technology using these techniques and all. I want to go in the cancer genetics through molecular diagnostics way, like sequencing the cancer genes
Mhm.
in the cervical cancer. So how should I approach for the companies? Can I do this project in
Yeah.
intern?
Actually, I think this question, the answer of this question would be very long. So you can just, I think at the end of this presentation, I will ask HR to share my email ID to you, or then you can write me there to ask your
Okay.
question. I will give you the very detailed answer over there. Okay.
Yeah, surely. Thank you, sir. It's a
Yeah.
great session. Sir?
Hello. Hello.
Yeah.
Hello. Good evening, sir. Myself Akshansh Kumar,
Mm-hmm.
and I'm from India, and I'm currently studying pharmaceutical biotechnology, M.Pharm. For getting internships in any college in abroad like Germany and all, sir, how we should approach, and for colleges or industry also, can you just
Okay.
guide?
Okay.
How can we approach?
Yeah,
Because
I got your point. I think this question is very related to the career thing. So as we have only, let me check, only 10 minutes left, and I can say I have different what I can say here, that because answer of this question will be long. So if you once you will complete your masters in biotechnology or any of the field you're working, then you have to apply for the for doing the, you want to join PhD, right?
yes, sir. Yes, sir. If
Yeah,
in
after doing your
continuously.
masters, if you want to join the PhD in Germany or in any of the European countries, especially I will tell you the European country, I don't know much about the USA. So
Yes,
if
sir.
you
Yes.
want to do the PhD over here, then again, you should, there are some basic requirements you need to be complete in your master degree. So when you do the masters, when you're doing the internship at the end of in the end semester of your master degree, so your this internship should be a productive. In the productive, in the sense you should have experience in a specific area. If, for example, if you want to join like pharmaceutical, then you have to do some productive internship in the pharmaceutical industry. If you want to join like biotechnology lab or immunological lab, then you should complete your internship in some of the nice institute of immunology, where they work on immunology or where they work on some other bioinformatic or biotechnology techniques. So, and the other most important thing is what nowadays selection committees were considering, that you should and you must have at least one publication in your master degree level. So try to transform your internship into the publication. This is the most important thing over here. So once you complete your masters, then there are 2 sessions in the year in Germany where you can apply. One is winter session and one is summer session. So now winter session is over, and next year the summer session will come. Then you have to apply with the different universities. Yeah.
Sure, sir. Sure, sir.
Yeah, actually, if you need detailed information about this, just write me, okay?
Yes, sure, sir.
Yeah.
Thank you, sir.
Yeah, welcome.
Thank you, sir.
So I can see in the chat there are some questions. So I'm going to reply there quickly. If there is any other question, you can ask me from the audience.
Hello, sir.
Yeah.
Sir, I'm from India, and I wanted to pursue my PhD in bioinformatics, but I don't have any prior experience in informatics and any knowledge regarding that. So, what the circumstances will I have to face?
Okay. So there are several things in bioinformatics which you need actually if you want to choose this bioinformatics as a career. So first one is that you should have some basic experience in the computer language. If you don't have, then you have to learn it. You have
Okay.
to learn it. You have to learn at least one computer language like Python. I prefer Python or R. And to learn this language, you have to choose some specific courses like job-oriented courses or some research-oriented courses. I think the Doctoromics, they also provide these type of courses. You can contact to them, and hopefully they will help to you. Yeah.
Okay, sir. Thank you.
Yeah, welcome.
Sir, I recently complete my master's and I complete my hello.
Hello, yeah. Ask your
Yes, sir.
question.
I recently complete my master's in bioinformatics and I done my internship with Biotechnica. So in bioinformatics. So how can I apply for PhD in Germany and European countries?
Okay, I think the major question is regarding to applying the PhD in Germany. So, as I have answered right now, that there are some of the like major things you need in your master degree to apply the PhD in foreign countries, especially Germany, USA, or Europe. So that thing is that you should have a like very keen knowledge about this programming language, which is must right now. You should have at least one publication published at your master degree level,
I
specifically
have
from
done my publication.
do you have, sorry?
I have done my publication and internship also.
Okay, if you have completed your publication and internship, then I will suggest you to keep your eye open on the advertisement of summer session and winter session. And if you need more detail how you can apply for these PhD, then can you please write me, because I will prepare some of these things and some links and then share with you. Okay.
Hello, sir. How are you doing?
Yeah, I'm good. Thank you.
Sir, my name is Nadia. I'm from Pakistan, and please share your email address.
Okay, I'm sharing my email address now. Okay, you can write me anytime, and I'm also sharing you my LinkedIn, and so you
Good
can.
evening. Okay. Good day.
Thank you.
Good day, sir. Sorry,
Yeah.
can you hear me?
Yeah, I can hear you.
Okay. I'm Godwin Vivian, and I'm from Nigeria. So I completed my master's in September this year under biotechnology in South Africa, and I had worked on RNA sequencing data from animals. Like, it was an experimental study. So I'm a little bit, should I say, a bit confused. This is more of like a personal question, because currently I do not have a strong work experience, because I had just left my undergrad honors and then straight to my master's. So I did hear you mention about internship, although where I did my master's studies, because it was a viral RNA studies, was in a sequencing firm which I had stayed there for 6 to 8 months, and I also struggled with my sequencing data because I initially didn't have any idea of programming language and all that. It was a whole lot. So now my question is, I'm really very confused whether I should go looking for an internship or probably a job, or go for my PhD. I got an offer for PhD still here in South Africa, but at the same time, I'm just trying to know what exactly is actually right for me now. It's easy to get a PhD here because I'm already still here in South Africa, and I have a good relationship with my supervisors. But at the same time, should I still continue in doing human genomics, and while maybe I could go get a real life experiences of job before maybe considering that? I don't know what counts for me at this point, and I'm kind of
I understand your question. I think there is a very nice opportunity with this Dr. Omics, they are conducting some job-oriented bioinformatics courses and also the research-oriented bioinformatics courses. I think more this my colleague will tell you in more detail, where they will teach you the current technologies of the bioinformatics. They will teach you everything, and they will you can work on real-time data, and at the end they will also help you to publish a research paper, what you will do in your internship over there. So I think it will be the great, and more about these things, I think HR will tell you more about these, right?
Okay.
Yeah, but I'm sure that you will get very much help from the Dr. Omics if you will look into their website, and they in the different courses they offer. Okay, and those
Sorry for
courses are
interrupting
very
you.
real-time
I did not
courses.
receive your email ID.
Okay, thanks.
Yeah, welcome.
Sir, we didn't receive your email and LinkedIn ID in the
Okay,
chat box.
yeah, I think at the end of this presentation you will get it.
Sir, on the by screen sharing.
Yeah, I think HR will send you my email ID. Okay.
Okay, sir. Thank you so much.
Sorry for interrupting you all here. And Dr. Yogesh, I think we should take some technical questions now.
Yeah.
And some of them are already present in a chat box. For example, somebody asked that.
Am I able to reply there? Because I reply some of the answer.
Some people are asking about the languages that they should command to get successful career in NGS and something like that.
During the process engine generation sequence we perform.
Good evening, sir.
Yeah, good evening.
Sir, my question was also the same. Like, what all languages you prefer that we should learn at the first? Like, we are beginners. So, what all languages do you prefer that we should learn at the beginning?
So at the beginning, I think I suggest you to start with the R programming language, and then move to the Python, and then in like with R you can also learn the Python. So if you will start with learning the R programming language, then you will not feel like burden of learning this language. R is a very simple language. It contains a very like beautiful packages of bioinformatics. So you should start with R, I think, if you are a beginner. Okay.
Okay, sir. Hanji. Thank you.
Yeah. Why we need available data from our sample. Okay. So.
Hello, sir. Can you hear me?
Yeah, I can hear you.
I'm currently pursuing BSc Bioinformatics from India, and I have strong programming foundations too, and basic data science and ML. So, what do you suggest? What kind of projects should we make to pursue and excel in NGS and integrate data science or ML applications in it?
Yeah. So what you can do is, if you are mastering in doing machine learning, then the best would be to integrate the different omics data set together. This is the very booming type of things at the moment, that if you will be able to develop some of the technical or some of the pipeline which integrate the different omics data together, and then it will be very nice for you. For example, if you are working on a cancer biology, so you want to identify if a patient is responding for chemotherapy or not. If you are giving a drug to the patient, at some point it's very usual in the cancer patient, specifically in pancreatic cancer patient. If you are giving a drug to a cancer patient, at some time point they started they stop responding against those chemotherapy. So what is the reason behind that? That the patient is stop responding against the chemotherapy. So you can do the integration of different omics and data set. You can use single cell, you can use RNA bulk, you can also use the metagenomics, and by integrating these all three different omics data set, you can identify some of the reason, what are the reason, or what are the genes, or what are the microbes which are playing role in inhibition or in non-responder of this chemotherapy in these patients. So if you have a good skill, you can develop a pipeline, or you can also develop like different interactive tools. And using
Hello?
machine learning, yeah, you can also use if you have connections, and if I think there will be also available data set, but you have to dig more into the different biological databases. If you able to get some serum based assay data set from online different sources, then you can develop a classification model which can classify the responder and non-responder cancer patients to the chemotherapy by using the machine learning techniques. Yeah.
Hello, sir.
Mhm.
Hello, sir. Can you hear me?
Yeah, please ask question. Dipshika, how many minutes we have?
Please take up the questions. We can stress that to 7:15 or 20, I guess.
Okay. Okay. Please, if there is any question, you can ask me. Otherwise, you can
Hello,
write
hello.
me.
There are some questions in chat box, like about your
Okay,
research
I
that
think
you
there are
use.
so many questions. So I'm scrolling down and down. Dipchika, can you copy these question and then I can answer them? And later on, I will send the file to you. Is it?
Yeah, sure.
Because there are so many questions and so many chat. Which programming language? Okay, I think I have answered some of these questions regarding the programming language.
Hello, sir.
Hello.
Yeah.
Hello, sir. Hello.
Hello. Yeah.
Sir,
Please ask.
yeah, sir. Sir, I have done my courses on molecular dynamics simulations and docking. Plus, I have also done my bachelor project in 2D QSR modeling and using machine learning and all. So, what if how can we use my molecular dynamics simulations and docking, sir? And what project we can relate, sir?
So using the molecular dynamics simulation and drug designing, in silico drug designing means you have experience in silico drug designing basically, right?
Yes,
And
sir.
developing a QSAR model or 2D and 3D both.
2D and 3D, sir, both, sir.
Okay, so now what is your question exactly? Now what do you want to
Sir,
do?
means,
Or what are you
sir,
exploring?
sir, currently, sir, I have done my bachelor project in 2D QSR and 3D QSR in both, in bachelor's only, sir,
Mm-hmm.
in my final year. And, sir, I want to continue my project related to anything. And currently, I have also learned molecular dynamics simulations and docking. So, what type of project you just can suggest for that?
So this is a very
Any
broad
other
question
topic?
actually. So it depends upon your choice, which area you like. So if you have experience in silico drug designing, especially in 2D and 3D QSAR modeling, then you don't have to specifically select one thing. You can, but it's your choice. I will say personally, it's your choice. You want to be a cancer biologist, you want to work in a cancer, but being a in silico drug designer, you don't have a specific area that you will only design the drugs for cancer, or you will design the drugs for malaria or any other thing. Whatever challenges will come to you, you have to do that. But even though if you want to focus yourself, then you have to be you have to jump more deeper into one specific area, like for example, cancer, or in COVID, you can say. Then you have to also jump into the next generation sequencing, or more into the molecular biology of that. So if you select yourself of one confined area for research in drug designing, then you have to learn more. You have to go, you also have to need to learn molecular biology behind that particular area.
Okay,
So
sir.
at being at graduation level, I think if you will go to the master's level, then you will be more focused. Did you
Sir,
did
I'm
BTech
currently
or
in master's
MSc?
only. No, sir. I currently in B Farm I did my project, and now
Okay,
currently I'm Farm. I'm
okay, you
from
are.
NGS College of Pharmacy, Mysore, and currently pursuing masters in pharmaceutical biotechnology from
Yeah,
Mysore, India.
I think it will be good if you will choose either this drug designing or pharmacophore modeling, or if you are more expert in other area of machine learning, then you can go to the like toxicity modeling. And yeah, I think it's completely depend upon your choice which area you want to go for.
Thank you, sir. Thank you,
Yeah,
sir.
welcome.
Hi, sir.
Hi.
My name is Sasi.
Yeah, I can
Sir.
hear you.
Okay, sir. Can you hear me, sir?
Yeah, I can hear you.
Okay, sir. My question is, I'm done master in MSc microbiology, sir. But till now, I'm seeking for a job, sir. I didn't know about knowledge, didn't any knowledge about microbiology careers, sir. If any know about microbiology careers, please suggest this. Sorry for my English, sir.
Sorry for interruption, but here we have very specific short span of time, and Dr. Yogesh will address the technical questions as of now, because otherwise he will address such kind of questions only. So if
Okay, ma'am.
anybody has any technical question regarding any NGS technologies, please unmute yourself and ask the question.
Yeah, please.
Oh, okay, ma'am. Thank you.
Yeah,
Thank
you
you for
can
your
ask if you have any question from the presentation, or if you want to know more about, okay, how we can retrieve the data and how we can do, is it possible to do, is it possible to do the analysis only from the published data and to make it in the publication form or not? So as I have told you that it is possible. You can also retrieve the data from the published paper, from their different repository, and you can do your own analysis on the data. If you find something interesting which is not published or which is not addressed in the published paper, then you can give the reference of that paper and write and do your own analysis. And if you get something new which is not there in the previous paper, you can write your own manuscript and you can proceed that manuscript for publication. Or if you are more advanced in doing this bioinformatics analysis, what you can do, you can download, suppose if you are working in a cancer, and a specific cancer, if you're working in a colorectal cancer, what you can do, you can download the specific condition, a specific type of cancer data set from different publication. Suppose you are downloading the RNA expression, RNA sequence data from, you have downloaded RNA expression data from 10 publication, and you have huge amount of data with you. Then what you can do, you can do the atlas type of analysis. Atlas type of analysis means you are considering, you are integrating all the 10 of the paper, and after integrating, you are having like I can say like 20 samples or 50 samples, which could be considered as an atlas based analysis. So you have, if you have a bioinformatics skill, you can integrate the different publication data set and find out some of the genes, which are, for example, if you can find out some of the genes which are different in different conditions. Suppose particular genes, if particular genes is showing low expression in normal condition and it's going high, it's showing high expression in the disease condition. So you can report, you can identify that gene, you can go more deeper in the pathway analysis of that gene, and you can coin your own story that you have done the atlas-based analysis by integrating the 20 samples of colorectal cancer, and you are reporting that this genes is found to be responsible for causing the colorectal cancer from normal condition to the disease condition. So you can do this type of research and write your manuscript, and you can easily publish those. So this is the one question, and I think there is, okay, most of the questions are like career related. Okay, there's a technical 80% of the. Okay, I think
Latest questions are technical, Dr. Yogesh, if you want to take them up.
okay, let me go down then. Okay. Okay, should I write? Okay, so there is one question that they found out bacteria and yeast in the culture, and how to identify the type of bacteria and fungus. So for this, to identify the type of bacteria and fungus, you have to go for the sequencing of the samples, and it's called metagenome sequencing. I think it will not cost much, and once you will get the sequencing, if you will send this your samples to the sequencing company, they will also give you the analysis part, or but they will charge you. But if you only want the sequencing, then you can ask them to sequence your data, sequence your sample, they will sequence, and then you can analyze and identify the bacteria by yourself using bioinformatics tools. Yeah, and for these analysis, you can use, there is a very, I will write it. So you can, once you will get the sequencing data, you can use this tool, which is called data tool, and you can do, I think it will be good. Yeah, and then the question, how do we know if the data has any error or not? I don't get this question properly. Type of error, which error? Okay, then, okay, PhD scholar. Okay, they did the next generation sequencing. Okay, this question is related to the cost of the sequencing, which I don't know, which is very difficult for me to answer, because the cost in Europe and in Germany and in India, it's very different. What you can do, you can just go, you can contact to the company and get the quotation of yours. You can send them, okay, you have these samples and data, and what will be the cost of that? You can choose any of the company. What type of bioinformatics work is supposed to do on the marine? Yeah, hello.
After this, I said. Hello.
Hello.
Am I audible?
Yes, Satvik, you are audible, please.
Yes, you mentioned some of the companies who offers bioinformatics job.
Yeah.
But I wanted to ask one question. If someone have the background in microbiology, was working in vaccine manufacturing, and he have some expertise in bioinformatics, like Custom W, he can handle the blast and FASTA.
Yeah.
So are there any freelancing service we can provide, or any chances for freelancing that we can do in India itself?
Satvik, again, I'm saying the same thing. This session is regarding the technical questions. Otherwise, some technical questions will be left. And you can write down your question in the feedback form, and definitely we will answer you.
Okay, ma'am, no problem. Thank you so much.
Good evening, sir.
Yeah, very good evening.
Sir, actually, I went to a science fest yesterday and I have made a model on monoclonal antibody production. Sir, you must be knowing it. It is in immunology about the hybridoma technology in which we harvest monoclonal antibodies on a major scale. Sir, there was a visit by a bioinformatician, and he asked me one question, like, how can you produce monoclonal antibodies without sacrificing a mice? Because in that technology, we are sacrificing a mice for its spleen to get the B cells. Sir, he actually asked me ki, how can you do this? So I was unable to answer that, but he just give me a hint that for the antibody which you are producing for that antigen, particular antigen, like basically cancer antigens, you can take the sequence of the epitope and then generate a monoclonal antibody. Like, I couldn't understood that. So what was the procedure basically?
So to do it completely in silico, right?
Yes, sir. I
Okay.
couldn't understood like what he was saying. He was a bioinformatician, and he said that we can take the sequence of the epitope of the antigen, and you can create an antibody like the sequence.
Actually, if you have epitope of the sequence, you can get the antibodies, but
But sir, are they reliable?
I can't say. I don't have answer of this question if they are reliable or not, but yeah.
Okay, sir. Okay, thank you.
You're welcome.
So I guess, Dr. Yogesh, we should move to the next speaker.
Yeah, sure.
Thanks a lot. Thanks a lot for your valuable inputs, and the session was really the session was really good. And I guess that all the audience are enlightened. And I request everybody to write down their questions if they have any in the feedback form, and we will definitely reply to them.